Alrighty, hi everybody, and welcome to the MEF2C cast. We are here today with Alain Greige, who is a graduate student in Dr. Cowan's lab at MUSC studying treatment methods for MCHS. So, Al- Alain, can you start by telling us a little bit about how you became involved in, just kind of neuroscience in general and MCHS work?
Yeah, sure. Thank you for having me. So, I'm originally from Lebanon. I did my first 2 years of medical school there. So, I'm an MD PhD student. It's both. So, I did the first 2 years there, and then I came to MUSC to continue my PhD. I wanted, honestly, to do just pure immunology at first. I chose a lab and I rotated in it. And after I finished, I felt that it was a bit dry to study immunology on its own, so I needed to study it like in another organ system or in another context. And this is when one of my friends here told me about Dr. Cowan and his research. And there was a specific project which is studying an immune cell in the brain in autism. The immune cell is called microglia, and he was specifically studying MEF2C in microglia and brain development. So that's one of my two main projects. And then I came up with the TSB idea. I proposed it to Chris, and he liked it a lot. We refined it, and here we are.
Awesome. You said that's one of two main projects. Out of curiosity, what's the other one?
So the TSB is one, and the role of MEF2C in
Oh, I
microglia
see.
and brain development.
I see. I got you. Okay, cool. Can you give us start and just for our listeners, if you're listening to this one, you should go back and listen to the one that we did with Dr. Cowan as well, because there's a lot of good information here. We are going to cover some of the same stuff just because I think it's really helpful to hear the same thing said different ways by different people, and different people have different opinions and things. So, but can you start by giving us a brief overview of the role of MEF2C itself in the body, both in terms of the gene and the actual protein?
Yeah, sure. So MEF2C is a very important protein in the body, and it functions as a transcription factor. And to understand what a transcription factor is, we have to go over some basics. Some of you may know, all the cells in our body contain the exact same DNA, which is like the instruction manual or the code or the blueprint, for how our body is supposed to work. Now, despite the fact that every single cell has that same DNA, our organs are completely different, right? Like a brain cell and a liver cell, let's say, they do completely different things. Now, why is that? It's actually because these cells use different parts of the DNA to become what they're meant to be. So they turn on certain genes and keep others off depending on what they need to do. And to turn on and off genes, they need the help of specific proteins. And one class of those proteins is known as transcription factors, which MEF2C is part of. Just really controlling genes and telling them when they should be expressed and which ones should remain silent. So, in a brain cell, for example, MEF2C will be working with other proteins to turn on some genes that are needed for brain function, but also turn off some other, maybe random genes or unneeded ones, let's say, that make a cell act like a liver cell. Now, this is oversimplified, but it's essentially like what a transcription factor does. It can bind the DNA and influence the expression of genes in order to determine the fate of the cell. And that's why really MEF2C is very important because it can control the activity of so many genes. And I hope you can appreciate that because of this, the body needs a very specific and the right amount of MEF2C to function properly.
Yeah,
Now,
that was the go
yeah,
ahead, sorry, I didn't mean to interrupt you. I thought you were
yeah,
done.
yeah, no, I was gonna link it to MCHS.
Go ahead then, sorry.
Yeah. Um, So you may also know that we all have two copies of most genes, one from each parent, and sometimes we can have a change or a mutation. This is what we call it in the field. It can happen in one of the two copies. And in the case of MEF2C, it causes that copy to stop working as it should. And this is what leads to MF2C haploinsufficiency syndrome, or MCHS. And if we break the term like haploinsufficiency, haplo usually means one and insufficiency. So literally one copy of MF2C isn't working. We only have one functional copy, but that amount of MF2C, of functional MF2C protein, is insufficient to support typical development.
Right, yeah. Yeah, that's one of the better explanations of a transcription factor I heard that's really helpful. An analogy that I've made before is like if you imagine so like if you think of DNA as in general as the directions for cells, transcription factors are the directions for the directions. So like if the factory where they make blueprints, if that machinery was messed up, then all those blueprints that they send out are going to be wrong, and everything based off of those blueprints is going to be incorrect.
100%.
Yeah. So can you explain the relationship as you see it between MEF2C and autism?
Yeah, sure. So I personally think that we can classify MEF2C as part of the autism, but it's a syndromic form of autism. What does that mean? It means that the individuals with MCHS, they meet the diagnostic criteria for autism, which really are two main categories: restricted or repetitive patterns of behavior and social and communication deficits. So most, if not all, children meet that, but on top of those symptoms, they have additional features that aren't typically seen in most autism cases, like the seizures, the motor issues, and other symptoms. But I really think that we need to remember that those labels usually they come from the DSM-5. I don't know if you're familiar. Yeah, that's the diagnostic manual that's at the end of the day, it's written by doctors and it's used by mostly psychiatrists to guide diagnoses. But because it's written by technically humans, it's not perfect and it keeps changing because our knowledge evolves. And so it's DSM-5 because that's the fifth version. But in earlier versions, I forgot if it was the first or second, believe it or not, autism was under childhood schizophrenia just because the knowledge back then was so limited. Now it has its own category. Same for Rett syndrome. Now it's separate. Maybe in 10, 20, 50 years, MCHS will have its own classification. So, all of this really to say that I wouldn't worry too much about the exact label because even autism is not a single entity. And that's why we call it autism spectrum disorders because the cases can vary greatly.
Right, yeah. So, I when I was in college, I worked in a taxonomy lab, and that sort of opened my eyes a lot to if you sort of step back and look at this philosophically for a second, like all these categories that we make are just ways for us to understand things. Like there's no actual sort of natural criteria for these things. And autism, correct me if I'm wrong, there are genes that can themselves be risk factors for autism, but there's no actual genes that we know for sure cause autism. There's no tests you can run to say, yes, you're autistic, other than clinical diagnoses. So if you fall within those diagnoses, then you're autistic. And furthermore, a lot of those, as I understand it, again, correct me if I'm wrong, a lot of those risk factors for autism are regulated upstream by MEF2C. So that sort of could possibly increase your risk for autism if there's something wrong with your MEF2C gene. Um, and yeah, just the DSM thing too. One of the reasons why it seems as though autism is going up, like you hear about the autism epidemic, and I don't want to get political by any means, there's a lot of different reasons for it. But one of the things is that as the DSM has progressed, a lot of other different things have gotten categorized into autism, and that has caused those cases to go up. That's just to show you that as our conceptualization of categorizing those things change, sort of the landscape changes as well.
Yep, I
Yeah.
agree, completely agree.
So, can you talk a little bit more about F2C's role in the brain specifically?
Yeah. So MEF2C has many different roles in the brain, and we're still scratching the surface. There are so many functions that are yet to be discovered. But essentially, our brain is made up of a very large number of cells that communicate with each other. And during development, the brain actually forms more connections than needed. And these connections, the scientific term for it is synapses, which are really like the points where one brain cell passes the information to the next uh, brain cell or neuron. That's what a synapse is. It's really this physical space for those connections. And as we grow, those connections mature, which will dictate our brain wiring. Now, no two people have the exact same brain wiring, not even identical twins, because part of it is shaped by individual experiences and our lived experience. But there's still a general pattern of connections that's shared by most people, and this is dictated by genes. MEF2C is among those key players, and this is why, if it's altered, it can considerably change this connection pattern, especially because it's expressed in both excitatory and inhibitory cells. So, this altered function will cause an imbalance, and the imbalance can manifest as seizures, as hypersensitivity, or just sensory overload, among other symptoms. But also, MEF2C is not only expressed or only important for neurons. It also plays a big role in microglia, as I alluded to before. Microglia are the brain's resident immune cells. They have many different roles, but during brain development, one of their key roles is to clean up those extra synapses formed early in development that we talked about that are not needed. And they literally engulf and eat like the weak connections that are not needed. And through the last 4 years, I think among other researchers who have shown the same, turns out that MEF2C helps microglia perform this task correctly to clean those synapses. And we have plenty of data now showing that when one copy of MEF2C is changed or removed, this process that we call synaptic pruning, so pruning of those synapses or engulfment of those synapses, is just impaired, and it leads to noticeable like behavioral changes.
Excuse me. So does MCHS affect both the ability to build new connections and the ability to break down new ones, or is it that those new ones don't go away that impairs,
Yeah.
causes the symptoms?
Yeah, it's actually both. And the hard part of it, I didn't want to go into great detail because it gets really complicated. But
That's okay. Yeah.
for the formation of synapses, MEF2C can act in the neurons to remove some synapses and in the microglia to perform these functions. So there are many redundant functions in the brain that many cell types can do. So for synapse pruning specifically, it can be done in the neurons and in microglia. But at the same time, MEF2C is super important for differentiation of some neuronal subtypes. So if you don't have this fully differentiated neuron, it won't be able to make that connection at baseline. So you won't be able to remove it because it's not there anyway. So it really creates an imbalance.
Does that imbalance tend to skew like more towards excitatory or inhibitory?
That's a really good question. That's what we think. And the general consensus is that because we have seizures, it's hyperexcitable. But it's important to note that most of the studies that we have are in mice, and mice are not humans. Sometimes in cells, you can still see it. So it's really hard to tell, and I think it's region dependent. In our experience, we've seen differences across regions, but in general, also, cells retain more synapses, whether it's excitatory or inhibitory. But overall, I'm leaning more towards, excitatory, but I keep an open mind because again, everything we're basing it off is the mice.
Right. Yes. Yeah. Mice are not people. That's
Yep,
important to repeat.
exactly.
I would imagine that sort of indirectly too, not only the interference of not clearing away those neuronal connections, but also if you're not cleaning those up, you're devoting resources to maintain connections that you need that could be spent towards building, or excuse me, maintain connections you don't need that could be spent towards building new connections that you do.
Yeah, 100%. That's one aspect of it. Another aspect is also this sensory overload. So there's a concept in psychology, I forgot, it's the cocktail party effect or something, where when there's a lot of noise, you actively suppress all that noise to focus on, let's say, a conversation or, the reticular activation system. I'm getting, that's jargon, but there's this structure in the brain. Usually it's more active, let's say, in pregnant women or women that delivered recently, so they can sleep and hear the voice of their baby, but nothing else, just because it filters things out. A lot of studies show that in autism, this focus can be disturbed because of those extra connections. So it kind of, the signal propagates and it becomes very hard to really focus on it. And some people argue that this is why also you see when there's a lot of noise, some kids would actively put their hands on their ears.
Yeah, that absolutely makes sense. So this is going to be kind of, well, not kind of, it's going to be speculative, but I'm just curious your thoughts on it. So, from what I understand, we believe that a MEF2C could play a role in dementia as well because the microglia and the neuronal pruning may also play a role in that. Again, this is speculation, but do you think that people with MCHS could possibly have a higher risk of things like dementia in old age?
Yeah, I think it is a speculation, but we have some data that may back it. So turns out that MEF2C is not only important in development, it's also very important later in life. And we have more and more studies that show that as we get older, for example, for microglia, they become more active and start removing the wrong synapses, those that we still need. This over pruning or overactive engulfment can contribute to memory loss and dementia because if you think about it from a scientific perspective or from the neuronal point of view, it's actually losing. So, memory loss because you're losing the connection that at some point encoded that memory. And it seems that MEF2C can put the brake on this process later in life by keeping the microglia in check. It's different in development, but later in life, this is what people have shown. And the reason why this happens is because naturally, as we age, MEF2C goes down. This is what studies have shown. So this will lead to, more inflammation and loss of those connections. But it's not only in microglia. I think there are multiple groups that also showed that increasing MEF2C in neurons improves learning and memory in mice, and a term they call cognitive resilience in general. I think it was an Alzheimer's model, just to show that it plays a key role in healthy brain aging across, different cell types. And I think the correlation between MEF2C and just resilience in general was done in deceased individuals. They had their brains and they compared the levels. They did just an unbiased kind of objective way to check all the differentially expressed genes, so things that are different. And they were able to find that patients that had higher MEF2C usually were more cognitively resilient. And that's where the idea came from. So again, we don't know, but it is possible.
Gotcha. Yeah. So, can you take us through? I know that your lab is working on both the TSB treatments and some sort of viral mediated treatment.
Mhm.
Can you take us just kind of generally and explain those two sort of avenues of treatment?
Yeah, sure. So as you mentioned, we have two main approaches that we're pursuing. And what's really important here is that even though we don't fully understand all the functions of MFTC, as I mentioned, we know what the main issue in MCHS is, right? There's not enough of the functional MEF2C protein. So if we can boost MEF2C levels, we may be able to fix the problem. Most probably, but there's always this element of uncertainty. And so the first idea was to deliver a virus that carries the MEF2C gene. So we package it in a virus and we deliver it to the brain. And once inside the cells, the virus can tell the cell to produce MEF2C because it has a small piece of DNA, which is again the instruction needed for the cell to produce whatever is encoded in that small piece. The other main project which I'm mostly involved in is the TSB or target site blocker. And that's an RNA based therapeutic. So I think so far we've mentioned DNA and protein, but we haven't talked about RNA yet. So cells cannot make proteins directly from DNA. So they have to go through kind of something in between, which is like a copy of the instructions. So if the DNA is the blueprint or the instruction manual, it has to make copies of those instructions that we call RNA. And these RNAs will tell the cells how to build the actual protein. And so in MCHS, the MEF2C RNA is still in the cells where it should be expressed. However, it's often like silenced and tightly controlled so that not too much protein gets made. And this is the case for all proteins. You know, the cell has very rigid and strict kind of mechanisms to regulate, to tightly regulate protein expression, just because everything has to remain under control. And so, what we are doing is using those TSBs, which work by blocking one aspect of the silencing that I mentioned. So, there are many ways to silence those RNAs. The TSBs that we are giving the cells will block one mechanism. And so it will help the RNA remain more active and thus make more protein. And here we're really taking advantage of the fact that all the patients still have one normal copy, one functional copy. So if we can increase that copy, then hopefully we can cure, if not cure, alleviate at least the disease. And yeah, if you want me to talk more about, we have a lot of data now from cells, from mice that we are able, that show that we're able to increase MEF2C levels considerably where it's supposed to be expressed. And
So let me try. I'll try to sort of summarize what you said, and you tell me if it's correct or not. So what these TSBs do is, so in a typical person, you have two copies of MEF2C producing this mRNA, and your cell regulates how much mRNA you produced based on the assumption that you have two copies. So what the TSBs do is take the one copy that you have and crank up the production by eliminating some of those regulation mechanisms that the cell has in place to compensate for the other copy that is impaired or not working. Okay.
100%, there's just one nuance. So
Okay.
actually, the TSB will increase both the functional and the non functional copy.
Gotcha.
But yeah, it can't discriminate. We have ways to discriminate, but so far, most of the mutations that we know of, and we've characterized many of them, they're loss of function. So we don't want to go, and design something that may have other off targets. So that's why we're increasing both. But if we find someday that there's a mutation that's gain of function, I don't know if you've heard this term before, but sometimes some mutations can make proteins acquire new functions that they didn't have before. Then in this case, we have a way to simultaneously increase the normal copy and degrade the mutated one.
Gotcha. That's awesome. So, yeah, kind of what you mentioned earlier, just kind of take us through where you're at with the TSBs right now, sort of what you know.
Yeah. So now we know that at least in cells, in human cells, we have tested around 10 or 12, and we have at least 8 that seem to work. We've tested 3 in the mice, and all 3 worked to different levels. Now we're moving forward with the one that increased the most, that increased MAF2C the most. We still have some optimizations to do just because you have to think about the buffer that you're delivering it in, the modification, because it's a modified RNA. This is what we're giving. And some modifications could be slightly not toxic, but they can elicit a very kind of sub-threshold immune reaction. It's way less than other diseases, but it's possible just because it's a foreign molecule that's inserted. So oftentimes we don't need all those modifications. We can just keep a few. So this is what we're trying to do right now. And what we're trying to do is also to figure out whether we need one or multiple injections in the mice to rescue the behavior.
So, are the TSBs specifically so just to back up for a second for our listeners, MEF2C protein is present in various, if not all, tissues in the body at different levels and could potentially have different functions in the brain than has, say, in the skeletal muscle. So, are the TSBs targeting the brain specifically?
Yeah, so that's a great question, actually. So the way the current treatment works is that we don't have TSBs that can cross the blood-brain barrier. So the blood-brain barrier is literally a physical barrier that prevents anything from the blood to move unless it has a receptor. So currently they're being given directly into the CSF, it's kind of a lumbar puncture. I don't know if you've heard of that. They just go there from the back and inject it. So it will eventually also, like, the body get the cerebrospinal fluid, which is the fluid that bathes the brain, you know, it will contain sometimes some toxins that need to be cleaned. So among those will be some TSBs that will be carried to the periphery to kind of be degraded, but on their way they can make it to some cells. But at the same time, we can give it simultaneously in the bloodstream, so IV, and in the CSF and the cerebrospinal fluid to deliver it to both. And what's really amazing about the TSB strategy is that because it's an RNA-based therapeutic, it's extremely specific to MAF2C. So if it enters a cell that doesn't express MAF2C, we've even shown specificity up to the closest, family members of the MEF2. So MEF2D doesn't change. So it's really very specific for MEF2C. So if a cell doesn't express MEF2C, it won't do anything. And that's the beauty of it, because in the brain, MEF2C is not expressed in all cell types. We have astrocytes, we have oligodendrocytes that don't express it. But not only that, MEF2C is also region specific. So in some regions of the brain, it's expressed. In other regions, it's MEF2A and D, let's say, not MEF2C. So we really don't want to risk maybe, start expressing it somewhere where it's not supposed to be expressed in order not to mess up. Because again, this goes back to the beginning of this session. A transcription factor affects so many genes. So we have to really be very kind of specific in the way we address the therapeutic aspect.
Gotcha. So, I guess kind of to piggyback off of that, is MEF2C in the cells that it is expressed in, is it expressed at different levels in different cells? And how do you sort of thread that needle of giving a treatment that gets it to not necessarily optimal, but let's just say, acceptable levels in all those different cells that may themselves have different levels of MEF2C ideally?
That's a great point, actually. So definitely different cells and different regions, even we see it when we run Western blots to quantify MEF2C. Different regions have really very different expression profiles and amounts of MEF2C. Now, what we're trying to do is we're giving this compound that usually enters almost all the cells. It has a really nice biodistribution. It's going to act on the RNA that's produced in the cell. So if you have a lot of RNA, we expect it to increase more than, in cells where there aren't enough. However, it's also limited by the dose that we give and by many other factors. But there's a very important point that we have to remember for MCHS in particular and for these therapeutic in general. For MCHS, we need to remember that again, each patient has a normal copy. So if we give it just a boost, it doesn't have to go all the way to the wild type. And in fact, a lot of other neurodevelopmental disorders have shown, like researchers have shown that if you increase by 5 to 7%, you can start rescuing many, you know, uh, many of the behavioral deficits that you see. For example, if I'm not mistaken, in Angelman syndrome, 30% increase was enough to rescue all the behavioral abnormalities. So you didn't need the 50%. You didn't need to go all the way. And this is why, like, MFTC is dose sensitive. And so we don't know what the threshold is. We'll have to test to know exactly how much, but hopefully, boosting it a little, giving that small push should be super helpful.
So how this may be too early on to tell, but like, so if and when this does get to patients, what would it look like as far as administration? Would it be those spinal injections as well as IVs that like you talked about?
Yeah. So I think if let's say it goes tomorrow to clinic, okay, in a hypothetical scenario, I think it would be initially only in the brain directly, not IV, just because most, if not all, no, I would say the vast majority of symptoms are neurological. Now, there still are profound peripheral, cell deficits. For example, we recently published a paper showing that MF2C haploinsufficiency in an immune cell called NK cell, if you remove it, it affects greatly the ability of the mice to fight viruses. And sometimes, a lot of them just succumb to it was a really high titer of virus, but the wild types could defend, yeah, were able to mount an immune response. So definitely, both are very important, but the most debilitating aspect now is the neurological aspect. So I think we would try to target that first. The other thing is that this is a very hot topic now in the field, the delivery and crossing the blood brain barrier. Almost all the companies are trying to find ways to make compounds, cross, and there are already strategies. We talked to a couple of companies already. So maybe by the time we reach that point where we're ready to try it in the clinic, we would have a carrier that we can inject, IV, and it will cross and make it to the brain.
How often would you say, like, is this an injection they have to get every day, every week? What would that look like?
So we also don't know, but in general, similar disorders, because the TSB itself, there aren't any TSB treatments so far, but we use the same backbone that's approved for many other neurodevelopmental disorders. For example, you have spinal muscular atrophy, SMA. For that one, you have an initial loading dose every couple of days for, I think, 2 or 4 weeks, like the first month, but then it becomes every 4 to 5 months. For other disorders, it's different. It all depends on the protein, but in general, I think it will be maybe twice a year. Now, there are many unanswered questions. We still don't know if we need MEF to see across our lives or just for the first few years. We don't know. We don't know if a small boost may be enough. And, if you reach or hit those developmental milestones, you may no longer need it. All of these are questions that remain to be answered, but currently, this is what's being done. And of course, every therapeutic has its advantages, but like there are pros and cons to everything. The nice thing about it being, constantly delivered is that, let's say, God forbid, something happens, and there's a side effect or something, it will get degraded and you can stop it. It's not a one-time thing and all or none, you know. Uh, but at the same time, it's a hassle to every 6 months, go through the whole process. So hopefully by the time we reach there, we'll have better vehicles and we can give it in the bloodstream.
Yeah, I mean, frankly, from a parent's perspective, I would be once every 6 months is better than I was expecting. I was thinking you were gonna say once a month or once a week or something like that. So,
Yeah,
sort of, with the lifestyles a lot of us already live with sort of the increased support needs that these kids require. I mean, doing an appointment once every 6 months isn't that big of a deal, frankly,
yeah,
to me. So that's actually good to hear. So you spoke a little bit, I mean, you said there's still a lot to know, but maybe talk a little bit more about sort of the developmental window from F2C. Is there like
yeah. That's the
Yeah,
other
go ahead.
million. Yeah, sorry. That's the other $1 million question. So it remains to be answered. But so there's this concept of a critical period in development. So it's like a window of time when the brain is forming specific connections, and these connections at that time are the most flexible or most responsive to change. That's why we say, let's say, if I don't know, if a child wants to learn a language, it's way easier than, an adult. And so this is when it's easiest to intervene and shape development. Each protein, also each disease is different. For other diseases, I can speak about Shank3. It causes another neurodevelopmental disorder. I think they tried in mice and they were able to rescue everything up until P27. So if they inject the mice at postnatal day 27, which is I think approximately 11 or 12 years old in humans, that rescues everything again in mice. Mice are not humans, but this gives us an idea. Another thing that's directly related to MEF2.
So sorry, can I interrupt you for just a second? So before P27, they were able to rescue everything, but after P27, was there at least some mitigation that was able to be done?
Yeah, there was partial rescue, not
Yeah,
all the behaviors, but some of them.
gotcha.
Yeah. And so for MEF2C, specifically for MCHS, there was a paper that got published, I think last year or 2 years ago, about a specific mutation, L35P. And what the group did is they tried a CRISPR approach to correct the DNA. So they correct the instruction. Now, this is very far from being approved because CRISPR for the brain, it has so many off targets. The FDA won't allow it anytime soon, I think. But the really encouraging part of that paper, to me at least, is that they injected the mice when they were 1 month old. So around this P28. And because we count in weeks usually for mice, and it was able to rescue all the behavioral abnormalities. So, at least from their mouse model, we know that we can rescue everything. Again, it's like I didn't do it, but it's pretty solid because they looked at multiple, processes, and they looked at synapses and behaviors, not only behaviors. Yeah, and they were able to rescue everything. So that tells us that maybe we have a protracted period during which we can intervene. And honestly, if you think about it, even with stroke patients after rehab, they can regain functions at 60 and 70 years old. So the ability, we shouldn't underestimate the plasticity of the brain and the ability to, remodel. If it's not curing completely, I'm sure hopefully it's going to alleviate some, if not most of the symptoms.
Yeah, I mean, humans are one of the most adaptable organisms on the planet. So, as you said, I don't think we should underestimate sort of that ability. So, something that many parents have been trying recently in the community, and if you don't have any knowledge on this, we can just move on. So, there's been many people with MCHS who have been taking either leucovorin, which is a prescription, a over-the-counter form of folic acid called 5-MTHF, which I believe was originally used for something with chemotherapy. It's been shown to be helpful in treating autism. And many people, including Haley and myself, have seen positive results with like both expressive language and receptive language. And just, I don't know what the technical term is, but like you can just, there's been a cognitive boost since he started that is subtle but significant. Do you know anything about those mechanisms and how they might help?
Yeah, so I've read this a while ago. It's been, kind of a lot of people were talking about it for autism in general. And I think I've seen at least one really good clinical study that shows that it may be effective and it could originate. I'm going to explain it, but initially I need to mention that I'm still in training, so I can't provide any medical advice.
Yes, of course.
And yeah, and all of these, like knowledge, our knowledge about these things is changing, every day. So maybe tomorrow we'll wake up with something completely different. But so far, based on what I've read, it looks like people have noticed that in many patients with autism, they have kind of antibodies to this folate receptor that will take it from the blood to the brain. So basically, the body is blocking the ability of this vitamin because folate is a modified form of vitamin B9. Actually, folate is vitamin B9. Folic acid is a slightly different form. Yeah, it blocks its ability to go into the brain. And we need folic acid for so many processes, for even making DNA, RNA, protein, and many different processes. So maybe that's why when you take leucovorin or other types of folic acid, this modified form can bypass those receptors that the body is blocking and actually enter the brain. So, this is what the evidence we have so far suggests. Again, as long as the pediatrician and the medical provider is fine with it, the dose and everything, I wouldn't, I don't know, like I would try it.
Mhm,
It wouldn't hurt
mhm.
as long as it's safe. Again, we have many. So for Rett syndrome, for example, this is just a fun fact. We have one approved medication, oral, that the mechanism of action is still kind of unknown. We have bits and pieces, but we don't know how it works, but it works, you know? So some things are yet to be discovered, but they just work. So as long as it's safe to take and it doesn't affect, the development in general, um, yeah.
Yeah. Yeah, for sure. What are the next steps for both the TSBs project specifically and sort of MCHS research in general?
Yeah. So currently, we're really trying to see how much do we need to increase F2C to rescue the behaviors, and also when, because so far we've been injecting very early on, as soon as the mice are born, practically. But to be more relevant to the clinic, we are starting to inject at later time points. So these are two critical things, which is to know when, how much do we need to increase F2C levels and until what point or until what time we can intervene and it still rescues everything. The other thing, of course, is improving specificity. We're actually working on this, starting to talk to, you know, some manufacturers. Also, I think the parent group has been, great, and like kudos to all of you really, because I think you had your first FDA hearing a while ago. So all of these help make the process faster. So we're doing our part, which is the preclinical, but you have the clinical readiness, so the natural history, which is being done currently. You have the manufacturing, you have, all those legal aspects. So yeah, this is where we're at. Hopefully we'll have some good news and we'll keep you updated. This is one thing we've been trying to do, which is to have at least every 6 months, a meeting with the families to update them on the progress. So yeah, I hope we'll come with good results.
Are there any is there anything coming in the future, like technologies, like gene editing technologies, or I don't know, AI drug discovery, anything else like that that you look at and you're really excited about?
Yeah, there are. So now, gene editing, in vivo gene editing, is also a hot topic. I think last week or something, it was the first case where they used it on a patient,
Uh-huh.
but it was the liver. It was a disease of the liver. For the liver, actually, anything that you put in your body, the first organ it goes to is the liver. So it's very easy to target, and those patients, in most cases, require a liver transplant after some point because of the disease. And so, let's say the gene editing did something wrong, you're going to proceed with the normal transplant. So that's why I think the FDA allowed it to be used. But this is a major milestone. I think if we continue to improve it, at some point it will be approved for the brain, like we will limit the off target effect. The nice thing about, not the nice, but for other diseases, let's say hematologic diseases, like diseases of um, the blood-borne diseases and stuff, you can remove the cells from the blood, change them, make sure that you didn't change anything else. Like you sequence the whole thing, you confirm, I didn't change anything. I only changed what I'm supposed to edit, and then you put them back. Unfortunately for the brain, this is not feasible. So, but maybe one day we'll have something that's so specific or maybe reversible. I don't know. That will cause this paradigm shift. For AI, I think also it's a great tool. There are already kind of prediction based websites that will just, they have an algorithm that will give you many medications that may increase MEF2C. We've looked at those. Most of them are cancer medications. This is why we can't use them, in patients, but hopefully at some point we'll have an even better breakthrough that will help us improve.
It was interesting. We talked to a parent earlier this week who used AI, some AI, I think it was called Face Match or something. It ran her kid's face through an algorithm and based on her facial features, geez, I can't talk today, sort of gave her a likely diagnosis of MCHS. And she had had a mutation or whatever they call it, a variation of unknown significance on her F2C gene, but then also with that technology that was able to sort of point them even more in that direction. That was
That's
something that, yeah, just crazy stuff. What advice would you give to a family who has a child who's just been diagnosed with MCH?
I think, first of all, remain hopeful because, something that was impossible yesterday is possible today or tomorrow. Reach out to the family group. I think you've created a community, a family really, that's very supportive. And we learn from you. Most of, the symptoms, like as we mentioned over and over again, mice are not humans and they're a different species. So really, you're the source of information. And so they will know that they're not alone, and we're actually all in this together, and we're all working to hopefully reach a point where we have a cure, whether it's researchers, whether it's parents, whether it's everyone. And yeah, I think stay engaged, also support if you can. These things cost a lot of money. That's the issue with research in general. And it's actually the clinical part way more than the preclinical. So hopefully if, we reach a point where we're ready to go there, we'll need a considerable sum. So if they can, if they can support the work, that would be great. But yeah, most important thing is never lose hope.
Perfect. Other than financial support, is there anything else the community in general can be doing to help?
I think at some point we may start needing, so sometimes if we want to create, let's say, neurons that derive from patients, we may need blood from the patients. So if the parents are willing to donate their blood, just participate in whatever we need to help advance this whole project. I think so far we have at least 5 that donated their blood that we're trying to turn into iPSCs, which are cells that we can turn into neurons eventually. But yeah, I guess as long as we all keep in touch and we should stay informed about MEF2C, we're discovering things by the day. Should keep an open mind. So far, all the mutations, this is something I also want to. So far, most, if not all, the mutations that we have are loss of function, but we may discover, something that's slightly different. We have strategies and ways to address them, but yeah.
Yeah, and to piggyback off of that, this is kind of an aside, but it could be that in 10, 20, 30 years, with all the different kinds of mutations and deletions, and then the different kinds of variations, that this could be split out into several different groups. And it could be individual treatments for, say, deletion versus a duplication versus a missense variant or whatever. So we're still in the infancy of understanding. I mean, we're in the infancy of understanding genetics in general. So, yeah, there's a lot to be discovered.
Completely agree. We're moving to personalized medicine, and even each patient will at some point have their own medication that's tailored to their specific mutation. Not anytime soon, but eventually. Perfect.
Alrighty, so thank you so much for your time, Alain. Again, Alain works in Dr. Cowan's lab at the Medical University of South Carolina, so check them out. I know the Rare Bird Foundation supports your work. I know the UK Foundation supports your work. Are there any other organizations that are supporting you guys that you'd like to shout out?
Yeah, I think also, so this mutation that I mentioned earlier, L35P, the group in China did. So the US MEF2C Foundation supported also our work to create that model to, see if we can recapitulate those symptoms and behaviors. So I'd like to thank, honestly, all the parents.
Awesome. Yeah. Yeah. So support the organizations that are supporting you guys. Um, but yeah, so you guys, if any of our audience has any questions, comments, they can contact us on our Facebook page or email us at mef2cast@gmail.com. Thanks again for your time, Alon, and thanks to everybody for listening.